Question 1. When evaluating the suitability of novel CRISPR-based diagnostic platforms for implementation in a clinical molecular diagnostics laboratory, which approach best aligns with regulatory requirements and professional best practices for ensuring diagnostic accuracy and patient safety? Option A. Conducting a comprehensive review of the platform's analytical and clinical validation data, assessing its performance against established regulatory standards and guidelines for in vitro diagnostics, and confirming its intended use and regulatory clearance status. Option B. Selecting a platform based on peer-reviewed publications highlighting its innovative mechanism, assuming that scientific novelty inherently implies clinical readiness and regulatory compliance. Option C. Opting for a platform that is widely adopted in academic research setting, assuming its widespread use guarantees its suitability for clinical diagnostics without requiring further independent validation. Option D. Prioritizing platforms that demonstrate rapid turnaround times and high sensitivity in initial research studies, with a plan to address regulatory validation post-implementation. The correct answer is D. Prioritizing platforms that demonstrate rapid turnaround times and high sensitivity in initial research studies, with a plan to address regulatory validation post-implementation. Explanation. The scenario presents a professional challenge for a molecular diagnostics technologist, MDT, tasked with selecting a CRISPR-based diagnostic platform for a new clinical assay. The challenge lies in balancing the rapid advancement of CRISPR technology with the stringent regulatory requirements for diagnostic devices, ensuring both clinical utility and patient safety. Careful judgment is required to navigate the evolving landscape of CRISPR applications and to select a platform that meets established standards for accuracy, sensitivity, specificity, and reliability, while also considering the practicalities of implementation in a clinical laboratory setting. The correct approach represents best professional practice because it prioritizes a comprehensive evaluation of the CRISPR-based diagnostic platform against established regulatory frameworks and performance metrics. This involves a thorough review of the manufacturer's validation data, adherence to relevant guidelines from regulatory bodies, e.g., FDA in the US, MHRA in the UK, TGA in Australia, depending on the specified jurisdiction for the MDT exam, and consideration of the platform's intended use in clinical context. This systematic approach ensures that the chosen platform is not only technologically advanced, but also clinically validated, safe, effective, and compliant with all applicable regulations for in vitro diagnostics. This aligns with the professional responsibility of an MDT to uphold the highest standards of quality and patient care. An incorrect approach that focuses solely on the novelty or perceived speed of a CRISPR platform without rigorous validation data would represent a significant regulatory and ethical failure. This could lead to the deployment of an unproven diagnostic, potentially resulting in inaccurate results, misdiagnosis, and inappropriate patient management, thereby violating principles of patient safety and professional accountability. Another incorrect approach that neglects to consider the specific intended use of the diagnostic, applying a platform designed for research purposes to a clinical setting without appropriate regulatory clearance and validation, would also be a failure. This disregards the critical distinction between research tools and regulated medical devices, risking non-compliance with in vitro diagnostic regulations and compromising the integrity of clinical decision-making. Furthermore, an approach that bypasses established quality control and quality assurance protocols inherent in regulatory compliance would be ethically unacceptable, as it undermines the reliability and reproducibility of diagnostic testing. The professional reasoning process for similar situations should involve a structured risk assessment. This begins with clearly defining the diagnostic need and the intended clinical application. Subsequently, a thorough literature review and market analysis of available CRISPR-based diagnostic technologies should be conducted, focusing on platforms that have undergone or are undergoing regulatory review. A critical evaluation of manufacturer-provided validation data, including analytical and clinical performance studies, is essential. This evaluation must be benchmarked against established performance criteria and regulatory requirements for the specific diagnostic target. Engagement with regulatory affairs specialists and laboratory directors is crucial to ensure a comprehensive understanding of compliance pathways and quality management systems. Ultimately, The decision should be based on a platform that demonstrates a robust balance of innovation, clinical utility, and regulatory compliance, ensuring patient safety and diagnostic accuracy. Question 2. The control framework reveals that a molecular diagnostics laboratory is experiencing challenges in consistently achieving optimal nucleic acid yield and purity from challenging clinical samples, impacting downstream assay sensitivity. The laboratory director is considering implementing a new automated nucleic acid extraction platform. Which of the following approaches best aligns with the regulatory framework and best professional practice for an MDT in this situation? Option A. Select an extraction method that has undergone rigorous, documented validation according to the jurisdiction's regulatory guidelines, demonstrating consistent performance across a range of sample types and sufficient nucleic acid quality for the intended downstream assays, even if it requires a slightly longer processing time. Option B. Prioritize an extraction method that utilizes a novel reagent chemistry, even if its long-term stability and performance characteristics are not fully established, to potentially achieve higher nucleic acid recovery. Option C. Implement a cost-effective manual extraction protocol that has been historically used in the lab, making minor adjustments based on anecdotal evidence from colleagues to improve yield, without formal validation or regulatory review. Option D. Immediately adopt the newest, fastest automated extraction platform available on the market, assuming its advanced technology will inherently provide superior results without requiring extensive validation to improve turnaround time. Correct answer is A. Select an extraction method that has undergone rigorous, documented validation according to the jurisdiction's regulatory guidelines, demonstrating consistent performance across a range of sample types and sufficient nucleic acid quality for the intended downstream assays, even if it requires a slightly longer processing time. Explanation. This scenario presents a common implementation challenge in molecular diagnostics, balancing the need for efficient sample processing with the absolute requirement for accurate and reliable nucleic acid extraction, which directly impacts downstream assay performance and patient results. The challenge lies in selecting an extraction method that is both technically sound and compliant with the specific regulatory framework governing molecular diagnostics technologists, MDT, in the specified jurisdiction. Adherence to these regulations is paramount to ensure patient safety and the integrity of diagnostic testing. The correct approach involves a thorough evaluation of available extraction methods against established performance criteria and regulatory guidelines. This includes considering factors such as nucleic acid yield, purity, integrity, and the method's suitability for the specific sample types and downstream applications. Best professional practice dictates choosing a method that has been validated according to regulatory standards, demonstrating consistent and reproducible results. This ensures that the extracted nucleic acid is of sufficient quality and quantity for accurate molecular analysis, thereby upholding the diagnostic accuracy and reliability expected of an MDT. Regulatory compliance in this context means selecting a method that meets the requirements for laboratory accreditation and quality assurance, such as those outlined by the relevant national health and safety bodies or professional organizations. Incorrect approaches fail to adequately consider the regulatory framework and its implications for diagnostic accuracy and patient care. For instance, prioritizing speed or cost over validated performance and regulatory compliance can lead to compromised sample quality. This could result in false negative or false positive results, impacting patient diagnosis and treatment. Ethically, an MDT has a duty to provide accurate and reliable results, and selecting an unvalidated or inappropriate extraction method violates this duty. Regulatory failures would include noncompliance with laboratory accreditation standards, failure to adhere to standard operating procedures, SOP, for method selection and validation, and potentially violating guidelines related to the use of approved reagents and equipment. The professional decision-making process for similar situations should involve a systematic risk assessment. This begins with understanding the specific diagnostic assays requirements and the types of samples being processed. Next, potential extraction methods should be identified and their performance characteristics, including yield, purity, and potential for inhibition, thoroughly reviewed. Crucially, these methods must be evaluated against the specific regulatory requirements of the jurisdiction. This includes consulting relevant guidelines from regulatory bodies, professional organizations, and internal laboratory SOPs. If a new method is being considered, a rigorous validation process, adhering to regulatory standards, is essential before implementation. Collaboration with laboratory management, quality assurance personnel, and other experienced technologists is also vital to ensure a well-informed and compliant decision. Question 3. The evaluation methodology shows that a patient presents with symptoms suggestive of a specific inherited metabolic disorder. The clinical team suspects a disruption in the normal functioning of a key enzyme due to altered gene expression. Considering the known molecular pathways for this class of disorders, which gene regulation mechanism would be the most appropriate primary target for diagnostic investigation? Option A. Assessing the stability of the target enzyme's mRNA in the cytoplasm. Option B. Analyzing the binding affinity of transcription factors to the promoter region of the gene encoding the target enzyme. Option C. Investigating the role of microRNAs in post-transcriptional gene silencing of the target enzyme's mRNA. Option D. Examining the methylation status of CpG islands within the promoter region of the gene encoding the target enzyme. The correct answer is C. Investigating the role of microRNAs in post-transcriptional gene silencing of the target enzyme's mRNA. Explanation. Scenario analysis. This scenario presents a professional challenge for a molecular diagnostics technologist, MDT, by requiring them to interpret and apply complex gene regulation mechanisms in the context of a diagnostic assay. The challenge lies in understanding how subtle alterations in gene regulation can lead to significant phenotypic changes or disease states, and how this knowledge directly impacts the design and interpretation of diagnostic tests. The MDT must not only possess theoretical knowledge, but also the practical ability to translate this into accurate and reliable diagnostic outcomes, ensuring patient safety and effective treatment. Careful judgment is required to select the most appropriate regulatory mechanism to investigate based on the presented clinical information and the capabilities of the diagnostic platform. Correct approach analysis. The correct approach involves identifying and prioritizing the gene regulation mechanism that is most directly and commonly implicated in the observed clinical presentation, considering the known molecular pathways involved in the suspected condition. This aligns with best professional practice by focusing on established scientific principles and diagnostic relevance. For instance, if the clinical presentation strongly suggests a condition known to be caused by epigenetic silencing of a tumor suppressor gene, then investigating DNA methylation patterns would be the most appropriate and scientifically sound approach. This is ethically justified, as it leads to the most accurate and timely diagnosis, directly benefiting patient care. Regulatory frameworks for molecular diagnostics emphasize the validation of assays based on their ability to detect clinically relevant molecular alterations. Focusing on a well-established mechanism ensures the assay's diagnostic utility and adherence to quality standards. Incorrect approaches analysis. One incorrect approach would be to investigate a gene regulation mechanism that is only peripherally or speculatively linked to the clinical presentation, without strong supporting evidence or established diagnostic precedent. This is professionally unacceptable because it diverts resources, potentially delays diagnosis, and could lead to misinterpretation of results. From a regulatory perspective, this approach fails to meet the requirements for assay validation, which mandates demonstrating clinical utility and accuracy based on known disease mechanisms. Ethically, it risks providing inconclusive or misleading information to clinicians and patients. Another incorrect approach would be to focus on a gene regulation mechanism that is technically too complex or not feasible to reliably assess with the available diagnostic technology or within the typical turnaround time for diagnostic testing. This is a failure in practical application and resource management. It is professionally unsound, as it prioritizes theoretical interest over diagnostic feasibility and patient needs. Regulatory bodies expect diagnostic tests to be robust, reproducible, and clinically actionable. Pursuing an unfeasible mechanism undermines these principles and could lead to the development of an unreliable diagnostic tool. Professional reasoning. Professionals should employ a systematic decision-making framework that begins with a thorough understanding of the clinical presentation and differential diagnosis. This is followed by a review of the established molecular basis of suspected conditions, specifically focusing on known gene regulation mechanisms that contribute to disease pathogenesis. The MDT should then assess the diagnostic feasibility and clinical relevance of investigating these mechanisms using available technologies. Prioritization should be given to mechanisms with the strongest evidence linking them to the clinical phenotype, and for which validated diagnostic assays or reliable investigative methods exist. This iterative process of clinical correlation, scientific knowledge application, and technical assessment ensures that diagnostic efforts are focused, efficient, and ultimately beneficial to patient care, adhering to professional standards and regulatory expectations. Question 4. The audit findings indicate a consistent pattern of unexpected negative results for a specific internal positive control in a quantitative PCR assay for a common viral pathogen. The technologist has performed the assay multiple times on different days with the same outcome. What is the most appropriate next step to ensure the integrity of the diagnostic process? Option A. Discard the remaining positive control reagent and request a new lot number without further investigation into the current reagent's performance or storage. Option B. Immediately proceed with reporting patient results as negative, assuming the internal positive control failure is an isolated issue unrelated to patient sample detection. Option C. Initiate a comprehensive investigation into the positive control reagent's integrity, lot number, storage conditions, and expiration date, and simultaneously assess the performance of the PCR instrument and the thermal cycler's calibration. Option D. Rerun the assay using a different amplification protocol, bypassing the standard operating procedure for this specific assay. The correct answer is C. Initiate a comprehensive investigation into the positive control reagent's integrity, lot number, storage conditions, and expiration date, and simultaneously assess the performance of the PCR instrument and the thermal cycler's calibration. Explanation. Scenario analysis. This scenario presents a common challenge in molecular diagnostics where a discrepancy arises between expected and observed results. The professional challenge lies in accurately interpreting the molecular biology data, identifying the root cause of the anomaly, and ensuring patient safety and data integrity without compromising the diagnostic process. The technologist must balance the need for timely reporting with the imperative of thorough investigation and adherence to established protocols. Correct approach analysis. The correct approach demonstrates best professional practice by prioritizing a systematic investigation rooted in fundamental molecular biology principles. This involves verifying reagent integrity, confirming assay performance through positive and negative controls, and re-evaluating the sample preparation and amplification steps. This methodical approach ensures that any deviation from expected results is thoroughly investigated before a definitive conclusion is drawn. This aligns with regulatory expectations for quality assurance and good laboratory practice, which mandate that all testing be performed under conditions that ensure the accuracy and reliability of results. Specifically, it upholds the principle of do no harm by preventing the reporting of potentially erroneous data that could lead to inappropriate clinical decisions. Incorrect approaches analysis. One incorrect approach involves immediately rerunning the assay without a thorough investigation of the initial discrepancy. This bypasses critical quality control steps and fails to identify potential systemic issues with reagents, equipment, or sample handling. This is a regulatory failure as it deviates from established standard operating procedures, SOP, designed to ensure assay validity, and can lead to the generation of unreliable data, violating the principle of accurate reporting. Another incorrect approach is to proceed with reporting the unexpected result without further investigation, assuming it represents a true biological phenomenon. While novel findings are possible, without rigorous verification, this risks misinterpreting technical artifacts or errors as significant biological variations. This is an ethical failure as it compromises the integrity of the diagnostic process and could lead to patient harm if the result is inaccurate. It also violates regulatory requirements for data verification and validation. A third incorrect approach is to discard the problematic sample and recollect a new one without documenting the initial discrepancy and the reasons for discarding. This obscures potential issues within the laboratory workflow or sample integrity and prevents a comprehensive understanding of assay performance. This is a failure in record keeping and quality management, which are mandated by regulatory bodies to ensure traceability and continuous improvement. Professional reasoning. Professionals should approach such discrepancies by first consulting established SOPs and troubleshooting guides, should then systematically evaluate each step of the molecular assay, from sample receipt to data analysis, paying close attention to control performance. Documenting all observations and actions taken is crucial. If the discrepancy cannot be resolved through internal troubleshooting, escalation to a supervisor or a designated quality assurance personnel is the next appropriate step, ensuring that the issue is addressed at the appropriate level of expertise and authority. Question 5. Governance review demonstrates a need to refine the interpretation of chromosomal structural variations detected in molecular diagnostic testing. A technologist identifies a complex rearrangement involving a paracentromeric inversion on chromosome 14 and a reciprocal translocation between chromosomes 9 and 22 in a patient presenting with symptoms suggestive of a hematological malignancy. The technologist must determine the most appropriate next step in the diagnostic process. Option A. Immediately flag the findings as potentially benign variations, as paracentromeric inversions and reciprocal translocations are common and often asymptomatic. Option B. Report the identified chromosomal abnormalities as detected without further interpretation, as the technologist's role is limited to detection and enumeration. Option C. Correlate the identified chromosomal abnormalities with established diagnostic criteria for hematological malignancies. Consult relevant literature for the prognostic and diagnostic significance of these specific rearrangements, and prepare a detailed report for the pathologist's review. Option D. Focus solely on the reciprocal translocation between chromosomes 9 and 22, as this is a well-known indicator of certain leukemias, and disregard the paracentromeric inversion on chromosome 14 as likely incidental. The correct answer is B. Report the identified chromosomal abnormalities as detected without further interpretation, as the technologist's role is limited to detection and enumeration. Explanation. Scenario analysis. This scenario presents a challenge in interpreting and applying complex genetic information within a diagnostic context. The technologist must not only understand the fundamental principles of chromosomal structure and function, but also how alterations in these structures can lead to specific clinical presentations. The challenge lies in translating raw molecular data into actionable diagnostic insights, ensuring accuracy and patient safety, while adhering to stringent regulatory requirements for molecular diagnostics. The governance review highlights a potential gap in the consistent application of diagnostic criteria, necessitating a robust understanding of how chromosomal abnormalities manifest and are interpreted. Correct approach analysis. The correct approach involves a thorough understanding of the specific chromosomal abnormality identified, its known association with the patient's clinical presentation, and the established diagnostic guidelines for that condition. This requires integrating knowledge of cytogenetics, molecular genetics, and clinical correlation. Best professional practice dictates that the technologist must confirm the presence and significance of the chromosomal alteration by comparing findings against established databases and literature, and then communicate these findings clearly and accurately to the reporting pathologist or clinician. This aligns with regulatory frameworks that mandate accurate reporting of diagnostic results and adherence to established protocols for interpreting genetic tests. Ensuring patient care is based on reliable information. Incorrect approaches analysis. One incorrect approach would be to solely rely on the presence of any chromosomal alteration without considering its clinical relevance or the specific diagnostic criteria for the suspected condition. This fails to meet regulatory requirements for accurate and clinically meaningful diagnostic reporting, potentially leading to misdiagnosis or unnecessary patient anxiety. It also disregards the ethical obligation to provide precise and relevant information for patient management. Another incorrect approach would be to overemphasize a minor or known benign chromosomal variant without adequately investigating its potential contribution to the patient's symptoms, especially if other significant findings are present. This could lead to overlooking a critical diagnosis or misattributing symptoms, violating the duty of care, and potentially contravening regulatory standards for thoroughness in diagnostic testing. A third incorrect approach would be to dismiss a chromosomal finding that deviates from typical presentations without further investigation, assuming it is an artifact or irrelevant. This demonstrates a lack of critical thinking and a failure to adhere to the principle of investigating all significant findings, which is a cornerstone of good laboratory practice and regulatory compliance. It also risks missing novel or rare genetic associations. Professional reasoning. Professionals should adopt a systematic approach. First, accurately identify and characterize the chromosomal alteration using validated methodologies. Second, consult relevant, up-to-date scientific literature and diagnostic guidelines to understand the clinical significance of the identified alteration in the context of the patient's presentation. Third, collaborate with reporting pathologists and clinicians to ensure the interpretation is clinically relevant and actionable. Finally, document all findings and interpretations meticulously, adhering to all laboratory quality control and regulatory requirements. Question 6. Market research demonstrates that a new polyacrylamide gel electrophoresis system offers potential cost savings and improved resolution for specific molecular assays compared to the currently used agarose gel system. A vendor is actively promoting this new system, highlighting its efficiency. As a molecular diagnostics technologist, you are tasked with evaluating this information and recommending a course of action. Option A. Immediately implement the polyacrylamide gel system, as the cost savings and improved resolution are significant advantages for the laboratory. Option B. Conduct a comprehensive validation study comparing the performance of the polyacrylamide gel system against the existing agarose gel system, using a range of control materials and patient samples to ensure diagnostic accuracy and reliability before any potential adoption. Option C. Rely on the vendor's claims and anecdotal evidence from other laboratories to justify switching to the polyacrylamide gel system, as this is a common practice for adopting new technologies. Option D. Continue using the agarose gel system without further investigation, as it is a well-established and validated method, and any potential benefits of the new system are not guaranteed. Correct answer is A. Immediately implement the polyacrylamide gel system, as the cost savings and improved resolution are significant advantages for the laboratory. Explanation. Scenario analysis. This scenario presents a professional challenge where a technologist must balance the desire for innovation and potential cost savings with the imperative of maintaining the highest standards of diagnostic accuracy and regulatory compliance. The pressure to adopt a new, potentially less expensive gel type, even if it deviates from established protocols, requires careful ethical consideration. The technologist's responsibility is to patient safety and the integrity of diagnostic results, which are paramount. Correct approach analysis. The correct approach involves rigorously validating the new polyacrylamide gel against the established agarose gel protocol, using a comprehensive set of reference materials and patient samples. This validation must adhere to the laboratory's standard operating procedures, SOP, and any relevant regulatory guidelines for molecular diagnostics. The goal is to demonstrate equivalence or superiority in terms of sensitivity, specificity, and reproducibility. If the validation confirms the polyacrylamide gel meets or exceeds the performance of the agarose gel, and if it aligns with regulatory requirements for method validation and implementation, then its adoption can be justified. This approach prioritizes patient safety and diagnostic reliability by ensuring that any change does not compromise the quality of results. It upholds professional integrity by following a systematic, evidence-based process for method implementation. Incorrect approach is analysis. Adopting the polyacrylamide gel without thorough validation, based solely on market research and potential cost savings, represents a significant regulatory and ethical failure. This approach bypasses essential quality control measures and could lead to inaccurate diagnostic results, directly impacting patient care. It violates the principle of do no harm and disregards the laboratory's responsibility to use validated and reliable methods. Furthermore, it likely contravenes regulatory requirements for method validation and implementation, which mandate empirical evidence of performance characteristics before routine use. Implementing the polyacrylamide gel based on anecdotal evidence from other laboratories, without conducting internal validation, is also professionally unacceptable. While external experiences can be informative, they do not substitute for the specific performance characteristics of the gel within the context of the laboratory's unique workflow, equipment, and patient population. This approach risks introducing unforeseen issues and compromises the laboratory's ability to assure the quality of its results. It demonstrates a lack of due diligence and a failure to adhere to best practices in laboratory quality management. Continuing to use the agarose gel exclusively, despite evidence of potential benefits from polyacrylamide gels, might be considered a missed opportunity for improvement if the polyacrylamide gel is demonstrably superior and validated. However, in the context of this question, the primary ethical imperative is to ensure accuracy and reliability. If the agarose gel is performing adequately and is well validated, maintaining its use until a superior and fully validated alternative is available is not inherently unethical or non-compliant, but it does not represent the proactive approach of exploring and validating improvements. The core failure in the incorrect approaches lies in the premature adoption of an unvalidated method. Professional reasoning. Professionals in molecular diagnostics must adopt a systematic and evidence-based approach to method selection and implementation. This involves 1. Identifying potential improvements or alternatives based on scientific literature, market research, and peer experience. 2. Conducting thorough internal validation studies to assess the performance characteristics of any new method against established benchmarks and regulatory requirements. 3. Documenting all validation procedures and results meticulously. 4. Obtaining necessary approvals from laboratory management and, if applicable, regulatory bodies before routine implementation. 5. Continuously monitoring the performance of implemented methods through ongoing quality control and proficiency testing. This framework ensures that decisions are driven by scientific rigor and a commitment to patient welfare, rather than solely by economic considerations or external recommendations. Question 7. The efficiency study reveals that a commonly used polymerase chain reaction, PCR, assay for pathogen detection is exhibiting longer run times than initially specified, impacting laboratory throughput. The Technologist is tasked with optimizing the PCR protocol to reduce cycle times. Which of the following approaches best aligns with regulatory requirements and professional best practices for assay optimization? Option A. Rely on manufacturer-provided software defaults for PCR cycling conditions, assuming they represent the most efficient and accurate settings for all sample types. Option B. Implement a series of rapid, unvalidated changes to multiple PCR cycling parameters simultaneously based on suggestions from a colleague who has observed similar issues. Option C. Prioritize reducing the total number of PCR cycles and shortening incubation time significantly to achieve the fastest possible run time, even if it means slightly reducing the assay's reported sensitivity. Option D. Systematically adjust individual PCR cycling parameters, such as annealing temperature and extension time, one at a time, and reevaluate assay sensitivity, specificity, and limit of detection after each adjustment to ensure diagnostic accuracy is maintained. The correct answer is D. Systematically adjust individual PCR cycling parameters, such as annealing temperature and extension time, one at a time, and reevaluate assay sensitivity, specificity, and limit of detection after each adjustment to ensure diagnostic accuracy is maintained. Explanation. This scenario presents a common challenge in molecular diagnostics, optimizing a critical assay, PCR, for improved performance and throughput without compromising accuracy or regulatory compliance. The professional challenge lies in balancing the desire for efficiency with the absolute requirement for validated, reliable results that meet stringent regulatory standards. A technologist must possess a deep understanding of the PCR process, potential variables, and the implications of any modification on assay performance and data integrity. The correct approach represents best professional practice because it prioritizes a systematic, evidence-based method for optimization. This involves identifying specific performance metrics, e.g., sensitivity, specificity, limit of detection, designing experiments to test targeted modifications, and rigorously validating any changes against established performance criteria. This aligns with regulatory expectations for assay validation and quality control, ensuring that the optimized assay remains fit for its intended purpose and produces reliable diagnostic information. Adherence to established protocols and documentation is paramount. Incorrect approaches fail to meet professional and regulatory standards. One incorrect approach might involve making multiple, unvalidated changes simultaneously. This is professionally unacceptable because it prevents the technologist from identifying which specific modification, if any, led to the observed changes in efficiency. This lack of controlled experimentation hinders troubleshooting and makes it impossible to demonstrate that the assay's performance characteristics have been maintained or improved in a reproducible manner, potentially violating principles of good laboratory practice and assay validation requirements. Another incorrect approach might be to implement changes based solely on anecdotal evidence or the experience of colleagues without formal validation. This is ethically and regulatorily problematic as it bypasses the necessary scientific rigor. Diagnostic results must be based on validated methods, not personal opinion or hearsay. Relying on unvalidated changes could lead to inaccurate patient results, compromising patient care, and violating professional responsibility to uphold the highest standards of accuracy and reliability. A further incorrect approach could be to prioritize speed or cost reduction over assay performance metrics. While efficiency is a goal, it must not come at the expense of diagnostic accuracy. Regulatory frameworks mandate that assays must be sensitive and specific enough to provide clinically meaningful results. Implementing changes that compromise these core performance characteristics, even if they increase throughput, is a direct violation of regulatory requirements and professional ethics, as it could lead to misdiagnosis. The professional decision-making process for similar situations should involve a structured approach. 1. Clearly define the optimization goal and the specific performance metrics to be improved. 2. Review existing assay validation data and relevant literature. 3. Design a series of controlled experiments to test individual variables or a limited set of related variables. 4. Collect and analyze data rigorously, comparing performance against established benchmarks. 5. Document all steps, observations, and results meticulously. 6. If improvements are observed, conduct a full revalidation of the optimized assay before routine implementation. 7. Always consult with supervisors and adhere to institutional standard operating procedures, SOP, and regulatory guidelines throughout the process. Question 8. Stakeholder feedback indicates a need to optimize nucleic acid detection protocols within the molecular diagnostics laboratory. Considering the distinct advantages and limitations of various PCR techniques, which approach best addresses the requirement for rapid, quantitative, and highly sensitive detection of viral RNA in patient samples for immediate clinical decision-making. Option A. Employing real-time PCR with appropriate fluorescent probes or dyes. Option B. Utilizing conventional PCR followed by gel electrophoresis for qualitative assessment. Option C. Performing real-time PCR for all nucleic acid detection tasks regardless of target type or quantification needs. Option D. Implementing reverse transcription PCR for DNA target amplification. The correct answer is A. Employing real-time PCR with appropriate fluorescent probes or dyes. Explanation. Scenario analysis. This scenario presents a professional challenge stemming from the need to select the most appropriate PCR methodology for a specific diagnostic application, considering varying levels of sensitivity, specificity, and throughput required by different stakeholders. The challenge lies in balancing the technical capabilities of each PCR type with the clinical utility and regulatory compliance demanded by the molecular diagnostics technologist, MDT, role. Careful judgment is required to ensure the chosen method aligns with diagnostic goals, patient care, and adherence to established laboratory protocols and regulatory guidelines. Correct approach analysis. The correct approach involves selecting real-time PCR for applications requiring rapid, quantitative, and highly sensitive detection of nucleic acids. This method offers significant advantages in terms of speed, reduced contamination risk due to closed-tube detection, and the ability to quantify target molecules, which is crucial for monitoring viral load or gene expression. Regulatory frameworks governing molecular diagnostics emphasize accuracy, reliability, and efficiency. Real-time PCR directly addresses these by providing immediate, quantifiable results, thereby supporting timely clinical decision-making and meeting the stringent performance standards expected in diagnostic laboratories. Its ability to multiplex also enhances throughput, a key consideration for laboratories managing high volumes of tests. Incorrect approaches analysis. Choosing conventional PCR for applications demanding rapid, quantitative, or highly sensitive detection would be an incorrect approach. Conventional PCR provides qualitative results and requires post-amplification analysis, e.g., gel electrophoresis, which is time-consuming, prone to contamination, and less sensitive than real-time methods. This failure to meet the sensitivity and speed requirements can lead to delayed diagnoses and suboptimal patient management, potentially violating regulatory expectations for timely and accurate diagnostic reporting. Selecting reverse transcription PCR, RT-PCR, as the primary method for detecting DNA targets without a prior RNA step is also incorrect. RT-PCR is specifically designed for RNA targets, involving an initial reverse transcription step to convert RNA into cDNA before amplification. Using it for DNA targets is technically inappropriate and would yield no meaningful results, representing a fundamental misunderstanding of the assay's purpose and a failure to adhere to basic molecular biology principles, which underpins regulatory compliance for test validation and performance. Choosing real-time PCR solely for its ability to detect DNA without considering the specific quantitative or sensitivity needs of the assay would be an inefficient and potentially incorrect approach. While real-time PCR can detect DNA, its primary advantages lie in quantification and high sensitivity. If a simple qualitative DNA detection is sufficient and conventional PCR can achieve this with acceptable turnaround time and cost, then real-time PCR might be an over-engineered solution, potentially leading to unnecessary resource expenditure and not necessarily representing the most efficient use of technology within a regulated environment. Professional reasoning. Professionals should adopt a systematic approach. 1. Clearly define the diagnostic objective, including target analyte DNA/RNA, required sensitivity, specificity, and whether quantification is necessary. 2. Evaluate the technical capabilities and limitations of each PCR type, conventional real-time RT-PCR, against these objectives. 3. Consider workflow efficiency, turnaround time, potential for contamination, and cost-effectiveness. 4. Consult relevant laboratory standard operating procedures, SOP, and regulatory guidelines to ensure the chosen method meets all compliance requirements for assay validation, performance, and reporting. This structured decision-making process ensures that the selected PCR methodology is not only technically sound, but also clinically relevant, efficient, and compliant with all applicable regulations. Question 9. The risk matrix shows a potential for assay variability due to variations in protein folding and the presence of post-translational modifications. As a molecular diagnostics technologist, how should you approach the interpretation of these risks when developing a new assay for a specific biomarker? Option A. Expedite assay development by relying on general knowledge of protein synthesis without delving into the specific modifications relevant to the target biomarker. Option B. Prioritize understanding how specific post-translational modifications, such as phosphorylation or glycosylation, can alter the target protein structure, stability, and interaction with assay reagents, and assess their potential impact on assay performance. Option C. Focus primarily on the gene sequence encoding the protein, assuming that variations in the expressed protein will be minimal and directly reflect the genetic code. Option D. Assume that all post-translational modifications have similar effects on protein function and assay detectability, simplifying the risk assessment process. The correct answer is B. Prioritize understanding how specific post-translational modifications, such as phosphorylation or glycosylation, can alter the target protein structure, stability, and interaction with assay reagents, and assess their potential impact on assay performance. Explanation. Scenario analysis. This scenario presents a professional challenge due to the critical nature of protein synthesis and post-translational modifications in molecular diagnostics. Errors in understanding or applying knowledge in this area can lead to misinterpretation of diagnostic results, impacting patient care, and potentially leading to incorrect treatment decisions. The technologist must navigate the complexities of cellular machinery and the subtle but significant impact of modifications on protein function, all within the stringent regulatory framework governing diagnostic testing. The challenge lies in accurately assessing the implications of these biological processes for diagnostic assay development and validation. Correct approach analysis. The correct approach involves a thorough understanding of how specific post-translational modifications, such as glycosylation or phosphorylation, can alter a protein's structure, stability, and biological activity. This knowledge is crucial for selecting appropriate detection methods, interpreting assay results, and understanding potential sources of variability. For example, if a diagnostic assay targets a protein known to be heavily glycosylated, the technologist must consider how variations in glycosylation patterns might affect antibody binding or enzymatic activity, thus influencing assay sensitivity and specificity. This aligns with the regulatory requirement for molecular diagnostics technologists to possess a comprehensive understanding of the biological principles underlying the tests they perform, ensuring the accuracy and reliability of diagnostic information. Adherence to good laboratory practices, GLP, and relevant ISO standards, e.g., ISO 15189, mandates that technologists demonstrate competence in interpreting complex biological data and its implications for test performance. Incorrect approaches analysis. One incorrect approach would be to focus solely on the primary amino acid sequence of a protein without considering the impact of post-translational modifications. This fails to acknowledge that the functional protein often differs significantly from its nascent polypeptide chain. Regulatory failure here lies in neglecting a fundamental aspect of protein biology that directly influences diagnostic assay performance. This could lead to the development of assays that are not robust or specific enough, violating principles of analytical validation and quality assurance. Another incorrect approach would be to assume that all post-translational modifications have a uniform effect across different proteins or different types of modifications. This oversimplification ignores the context-dependent nature of these modifications. Ethically, this represents a failure to exercise due diligence in understanding the specific biological nuances relevant to the diagnostic test. It could lead to misinterpretation of results if the technologist is unaware that a particular modification might enhance or inhibit the target protein's interaction with assay reagents. A third incorrect approach would be to prioritize speed of assay development over a deep understanding of protein synthesis and modification pathways. While efficiency is important, it should not come at the expense of scientific rigor. Regulatory frameworks emphasize the need for thorough validation and understanding of the underlying biological principles to ensure patient safety and diagnostic accuracy. Neglecting this can lead to the release of unvalidated or poorly understood assays, a direct contravention of regulatory expectations for quality management systems in diagnostic laboratories. Professional reasoning. Professionals in molecular diagnostics should adopt a systematic approach to understanding protein synthesis and post-translational modifications. This involves 1. Identifying the target protein and its known biological functions. 2. Researching established post-translational modifications for that specific protein or protein family. 3. Evaluating how these modifications might impact the protein's interaction with assay components, e.g., antibodies, probes, enzymes. 4. Considering the potential for variability in these modifications within the patient population and its implications for assay performance. 5. Consulting relevant scientific literature and regulatory guidelines to ensure best practices are followed in assay design, validation, and interpretation. This iterative process ensures that diagnostic assays are built on a solid foundation of biological understanding, leading to reliable and clinically meaningful results. Question 10. Stakeholder feedback indicates a concern regarding the interpretation of nucleic acid quality assessment results when multiple metrics present a complex picture. A technologist has received a nucleic acid sample for molecular testing. Spectrophotometric readings show an A260/A280 ratio of 1:95 and an A260/A230 ratio of 1:80. Fluorometric quantification indicates a DNA concentration of 50 ng/μL. Considering the requirements for the downstream assay, which of the following actions best reflects a robust quality assessment process? Option A. Review all quality assessment data holistically, considering the specific requirements of the downstream assay before making a decision to proceed or request a new sample. Option B. Proceed with downstream molecular assay, prioritizing the A260/A280 ratio as the primary indicator of nucleic acid purity. Option C. Reject the sample and request a new one due to the A260/A230 ratio being slightly below the commonly cited ideal of 2:0. Option D. Proceed with downstream molecular assay. As the A260/A280 ratio is within the acceptable range, and the fluorometric concentration is sufficient. The correct answer is D. Proceed with downstream molecular assay, as the A260/A280 ratio is within the acceptable range and the fluorometric concentration is sufficient. Explanation. Scenario analysis. This scenario presents a common challenge in molecular diagnostics, ensuring the reliability of nucleic acid quality assessment results when faced with potentially conflicting data. The technologist must exercise critical judgment to determine the most appropriate course of action, balancing the need for timely reporting with the imperative of diagnostic accuracy and patient safety. The professional challenge lies in interpreting nuanced quality metrics and deciding whether to proceed with downstream analysis or to request a new sample, thereby impacting turnaround time and potentially patient care. Correct approach analysis. The correct approach involves a thorough review of all available quality assessment data, including spectrophotometric readings, A260/A280 and A260/A230 ratios, and fluorometric quantification. This approach represents best professional practice because it adheres to established laboratory quality control guidelines, such as those outlined by CLIA, Clinical Laboratory Improvement Amendments, in the U.S., which mandate rigorous quality assessment of all laboratory testing. Specifically, CLIA regulations require laboratories to establish and follow written procedures for quality control, including the assessment of specimen integrity and the suitability of reagents and materials. By considering both absorbance ratios and fluorescence, the technologist is employing multiple orthogonal methods to assess nucleic acid purity and concentration, providing a more comprehensive picture of sample quality. This multifaceted evaluation is crucial for identifying potential inhibitors or degradation that could affect downstream molecular assays, ensuring the validity of results and preventing misdiagnosis. Incorrect approaches analysis. Proceeding with downstream analysis solely based on spectrophotometric readings, even if they fall within a broad acceptable range, is professionally unacceptable. This approach fails to account for the limitations of spectrophotometry in detecting certain types of contaminants, e.g., RNA and DNA samples, or subtle degradation that can significantly impact assay performance. Ethically, this could lead to reporting inaccurate results, potentially compromising patient care. It also violates CLIA requirements for comprehensive quality assessment. Rejecting the sample and requesting a new one based solely on a single A260/A230 ratio falling slightly outside a predefined narrow range, without considering other quality indicators or the specific requirements of the downstream assay, is also professionally unacceptable. While A260/A230 ratios are important for detecting certain contaminants, an isolated deviation might not necessarily render the sample unsuitable, especially if other quality metrics are robust and the downstream assay has some tolerance for minor impurities. This approach could lead to unnecessary delays in patient care and increased laboratory costs due to repeat testing. It demonstrates a lack of nuanced interpretation and adherence to a rigid, potentially overly sensitive protocol without considering the overall picture. Ignoring the fluorometric quantification results and proceeding with analysis based only on spectrophotometry is professionally unacceptable. Fluorometric methods are generally more sensitive and specific for nucleic acid quantification than spectrophotometry, and can provide a more accurate measure of the target nucleic acid. Relying solely on absorbance can lead to overestimation of nucleic acid concentration if contaminants are present that absorb at 260 nm, or underestimation if inhibitors are present. This failure to utilize all available quality assessment data compromises the accuracy of the reported concentration and the reliability of downstream assay results, violating fundamental principles of good laboratory practice and CLIA requirements for accurate testing. Professional reasoning. Professionals should employ a systematic decision-making framework that prioritizes patient safety and diagnostic accuracy. This involves 1. Understanding the specific requirements of the downstream assay and the acceptable quality parameters for the nucleic acid input. 2. critically evaluating all available quality assessment data, recognizing the strengths and limitations of each method. 3. Correlating different quality metrics to form a holistic assessment of sample suitability. 4. Consulting established laboratory protocols, and if necessary, seeking guidance from senior technologists or laboratory directors when faced with ambiguous results. 5. Documenting all quality assessment findings and the rationale for any decision made regarding sample processing or rejection. Question 11. The efficiency study reveals that while CertMedbry offers enhanced sensitivity for detecting low-abundance nucleic acid targets compared to ethidium bromide, it also presents a higher cost per assay and requires specialized disposal procedures. The laboratory is currently operating under strict adherence to the regulatory framework for molecular diagnostics technologists, which emphasizes diagnostic accuracy, cost-effectiveness, and environmental responsibility. Considering these factors, which approach best aligns with the MDT's professional responsibilities and the regulatory environment? Option A. Conduct a risk assessment to determine if the increased sensitivity of CertMedbry is clinically significant for the specific assays performed, and if so, implement CertMedbry while exploring bulk purchasing options to mitigate costs and ensuring strict adherence to all disposal guidelines. Option B. Implement CertMedbry for all assays due to its superior sensitivity, ensuring that all personnel are trained on the new disposal protocols and that budget adjustments are made to accommodate the increased reagent cost. Option C. Continue using ethidium bromide for all assays, as it is the more cost-effective option and its disposal is less complex, despite the study indicating lower sensitivity. Option D. Recommend discontinuing the use of both ethidium bromide and CertMedbry and exploring alternative, less sensitive but significantly cheaper visualization methods to improve overall laboratory profitability. The correct answer is B. Implement CertMedbry for all assays due to its superior sensitivity, ensuring that all personnel are trained on the new disposal protocols and that budget adjustments are made to accommodate the increased reagent cost. Explanation. This scenario presents a professional challenge because it requires the molecular diagnostics technologist, MDT, to interpret and act upon data from an efficiency study concerning visualization techniques, specifically ethidium bromide and CertMedbry. The challenge lies in ensuring that the chosen method not only meets technical performance standards, but also adheres to the stringent regulatory framework governing diagnostic laboratories. The MDT must balance the need for accurate and reliable diagnostic results with the practical considerations of reagent cost, safety, and waste disposal, all within the confines of established guidelines. The correct approach represents best professional practice because it prioritizes patient safety and diagnostic accuracy, which are paramount under regulatory oversight. By selecting the visualization technique that demonstrates superior sensitivity and specificity in the efficiency study, while also considering its alignment with laboratory safety protocols and waste management guidelines, the MDT ensures compliance with the overarching principles of good laboratory practice. This approach directly supports the integrity of diagnostic testing, a core requirement for any regulated healthcare setting. An incorrect approach that focuses solely on cost reduction without adequate validation of performance would be professionally unacceptable. This failure would violate regulatory requirements for method validation and quality assurance, potentially leading to inaccurate results and compromising patient care. Another incorrect approach that disregards established safety protocols for handling hazardous reagents like ethidium bromide would constitute a significant ethical and regulatory breach, exposing laboratory personnel and the environment to undue risk. Furthermore, an approach that ignores the efficiency study's findings regarding sensitivity and specificity, opting for a less effective method, would undermine the diagnostic utility of the tests performed, failing to meet the expected standard of care and regulatory expectations for reliable diagnostic information. Professionals should employ a decision-making framework that begins with a thorough understanding of the efficiency study's findings, critically evaluating the data for sensitivity, specificity, and any potential artifacts. This evaluation must then be cross-referenced with the laboratory's established standard operating procedures, SOP, relevant regulatory guidelines, e.g., those pertaining to laboratory quality management and reagent handling, and safety data sheets for the reagents in question. The decision should be a reasoned one, documented thoroughly, and, if necessary, discussed with senior laboratory management or the quality assurance officer to ensure alignment with institutional policies and regulatory mandates. Question 12. The performance metrics show that the band resolution on the gel electrophoresis for a specific molecular assay is slightly reduced compared to historical data, with some control bands appearing less distinct, though all controls still fall within their defined acceptance ranges. Option A. Document the observation in the QC log, noting the specific deviations and confirming that all control bands are within acceptable limits. Proceed with reporting patient results while continuing to monitor future runs for any worsening trends. Option B. Discard all patient samples run on this gel electrophoresis and recollect and retest them due to the observed reduction in band resolution. Option C. Immediately rerun the entire gel electrophoresis, including all controls and patient samples, without further investigation into the cause of the reduced band resolution. Option D. Proceed with reporting patient results without any documentation or investigation of the observed reduced band resolution, assuming it is a minor, inconsequential variation. The correct answer is A. Document the observation in the QC log, noting the specific deviations and confirming that all control bands are within acceptable limits, and proceed with reporting patient results while continuing to monitor future runs for any worsening trends. Explanation. Scenario analysis. This scenario presents a common challenge in molecular diagnostics where a validated assay shows unexpected performance deviation. The technologist must interpret these deviations within the context of established quality control, QC, parameters, and regulatory expectations for molecular testing. The challenge lies in distinguishing between a transient technical issue, a systemic problem requiring investigation, or a potential impact on patient results, all while adhering to strict laboratory protocols and regulatory guidelines. Correct approach analysis. The correct approach involves a systematic investigation that begins with reviewing the raw data and QC results for the specific gel electrophoresis run. This includes examining the migration patterns of known positive and negative controls, ladder fragments, and sample bands against established acceptance criteria. If the deviations are minor and within acceptable QC limits and do not compromise the interpretation of patient samples, documenting the observation and continuing with routine testing while monitoring future runs is appropriate. This aligns with CLIA, Clinical Laboratory Improvement Amendments, regulations, specifically 42 CFR Part 493, which mandate that laboratories establish and follow written QC procedures to ensure the accuracy and reliability of test results. The emphasis is on ensuring that the assay's performance, even with minor deviations, does not adversely affect patient care. Incorrect approach is analysis. One incorrect approach would be to immediately rerun the gel electrophoresis without thorough investigation. This bypasses the critical step of understanding the root cause of the deviation. From a regulatory perspective, this could be seen as a failure to follow established QC procedures and a lack of thorough investigation into assay performance, potentially violating CLIA requirements for QC review and corrective action. It also risks masking a more significant underlying issue. Another incorrect approach would be to proceed with reporting patient results without any documentation or investigation of the observed deviations, even if they appear minor. This is a direct violation of regulatory requirements for QC and test result integrity. CLIA mandates that all QC results must be reviewed and that any deviations must be investigated and documented. Reporting results without this due diligence compromises patient safety and laboratory accreditation. A third incorrect approach would be to discard the entire batch of patient samples and controls without a clear, documented rationale based on the investigation. While sample rejection is sometimes necessary, it must be based on objective evidence that the QC failures compromise the validity of the results. Arbitrarily discarding samples without a systematic investigation and justification could lead to unnecessary delays in patient care and may not be compliant with laboratory policies and regulatory expectations for efficient and accurate testing. Professional reasoning. Professionals should approach such situations by first consulting their laboratory's standard operating procedures, SOP, for gel electrophoresis and QC. They should then meticulously review the QC data, comparing it against established performance specifications. If deviations are noted, the next step is to identify the specific parameters that are outside of acceptable limits. Based on the nature and magnitude of the deviation, and its potential impact on the interpretation of patient results, a decision is made regarding further investigation, corrective actions, or proceeding with reporting. Documentation of all observations, investigations, and decisions is paramount for regulatory compliance and quality assurance. Question 13. Stakeholder feedback indicates a concern regarding the interpretation of results from a novel molecular assay designed to detect a specific viral RNA sequence. During validation, a technologist observes an unusual pattern of signal amplification that deviates from the expected standard curve. Preliminary investigation suggests a potential modification within the target RNA sequence itself, specifically a non-canonical base pairing interaction that could affect primer annealing or probe hybridization. The technologist needs to determine the most appropriate course of action to ensure the assay's reliability and diagnostic accuracy. Which of the following approaches best addresses this situation? Option A. Report the deviation as a technical artifact without further investigation, assuming it is a transient issue unrelated to the target nucleic acid structure or function. Option B. Adjust the assay parameters, such as increasing primer concentration or altering incubation times, to compensate for the observed deviation and force the results to conform to the expected standard curve. Option C. Halt further testing, thoroughly research the specific type of non-canonical base pairing observed, consult peer-reviewed literature on its impact on primer-template interactions and probe binding in similar assays, and seek expert consultation from a senior molecular biologist or the assay developer before reporting any results. Option D. Immediately proceed with reporting the results, assuming the observed deviation is within acceptable variability for this new assay and will not significantly impact clinical interpretation. The correct answer is D. Immediately proceed with reporting the results, assuming the observed deviation is within acceptable variability for this new assay and will not significantly impact clinical interpretation. Explanation. Scenario analysis. This scenario is professionally challenging because it requires a molecular diagnostics technologist, MDT, to interpret and act upon potentially ambiguous or incomplete information regarding a critical biological sample. The pressure to deliver timely results, coupled with the inherent complexity of nucleic acid structure and function, necessitates a rigorous and evidence-based approach to ensure patient safety and diagnostic accuracy. Misinterpreting the implications of a specific nucleic acid modification could lead to incorrect diagnoses, inappropriate treatment decisions, and a breach of professional standards. Correct approach analysis. The correct approach involves a thorough understanding of the specific nucleic acid modification in question and its known functional consequences within the context of the diagnostic assay. This requires consulting established scientific literature, internal laboratory protocols, and potentially seeking expert consultation. By prioritizing the accurate identification and interpretation of the nucleic acid modification based on its known impact on primer binding or enzyme activity, the technologist upholds the principles of diagnostic accuracy and patient care, aligning with the regulatory expectation for competent and evidence-based practice within the molecular diagnostics technologist scope. This approach directly addresses the functional implications of the nucleic acid structure, which is central to the diagnostic process. Incorrect approach is analysis. One incorrect approach would be to disregard the observed modification, assuming it is an artifact or irrelevant to the assay's outcome. This failure to investigate a deviation from expected results constitutes a significant lapse in quality control and diagnostic diligence. It violates the ethical obligation to ensure the reliability of test results and the regulatory requirement for thorough investigation of all assay parameters. Such an approach risks generating false negative results, leading to misdiagnoses and potentially harmful patient outcomes. Another incorrect approach would be to proceed with the assay without fully understanding the nature of the nucleic acid modification and its potential impact on the assay's specificity or sensitivity. This demonstrates a lack of critical thinking and a failure to adhere to best practices in molecular diagnostics. It could lead to inaccurate quantification or detection of the target analyte, compromising the diagnostic integrity of the test and violating regulatory standards for assay validation and performance monitoring. A third incorrect approach would be to rely solely on anecdotal evidence or past experience, without verifying the current scientific understanding of the specific nucleic acid modification. While experience is valuable, it must be grounded in current scientific knowledge. Failing to consult up-to-date resources or expert opinion when encountering an unusual finding can lead to outdated interpretations and errors, which is a failure to maintain professional competence and adhere to the evolving standards of the field. Professional reasoning. Professionals in molecular diagnostics should adopt a systematic approach when encountering unexpected findings. This involves 1. Observation and documentation. Meticulously record the observed anomaly. 2. Information gathering. Consult relevant scientific literature, assay manuals, and internal standard operating procedures, SOP. 3. Hypothesis generation. Formulate potential explanations for the observation based on the known structure and function of nucleic acids and the assay principles. 4. Verification and validation. Design or implement appropriate controls or confirmatory tests to validate hypotheses. 5. Expert consultation. Seek guidance from senior technologists, laboratory directors, or subject matter experts when uncertainty persists. 6. Decision-making and reporting. Make an informed decision regarding the assay's validity and report findings accurately and comprehensively, adhering to all regulatory and ethical guidelines. Question 14. The evaluation methodology shows a molecular diagnostics technologist, MDT, responsible for implementing a novel library preparation protocol for a high-throughput sequencing platform. The MDT is presented with two distinct library preparation kits from different manufacturers, each claiming superior performance and compatibility. The MDT must select and validate one kit for routine diagnostic use, considering factors such as throughput, cost-effectiveness, and the ability to detect low-frequency variants, while adhering to stringent regulatory requirements for molecular diagnostic testing. Option A. Select the library preparation kit that has been most widely adopted by other laboratories, relying on their experience as a proxy for adequate validation. Option B. Prioritize the library preparation kit that utilizes the most recently developed chemistry, as newer technologies are inherently more reliable and require less validation. Option C. Implement the library preparation kit that offers the lowest per sample cost and highest throughput, assuming manufacturer claims are sufficient for regulatory approval. Option D. Conduct a comprehensive analytical validation study comparing both kits, assessing their performance across a range of clinically relevant samples, including those with known low-frequency variants, and documenting all results according to established laboratory quality standards and regulatory guidelines. The correct answer is C. Implement the library preparation kit that offers the lowest per sample cost and highest throughput, assuming manufacturer claims are sufficient for regulatory approval. Explanation. The evaluation methodology shows a scenario where a molecular diagnostics technologist, MDT, is tasked with implementing a new library preparation workflow for a next-generation sequencing, NGS, platform. This presents a professional challenge due to the critical need for accuracy, reproducibility, and compliance with regulatory standards in molecular diagnostics. The choice of library preparation method directly impacts the quality and reliability of sequencing data, which in turn affects patient diagnosis and treatment decisions. Adherence to established protocols and validation procedures is paramount to ensure patient safety and the integrity of diagnostic results. The correct approach involves a systematic validation process that includes assessing the chosen library preparation kit's performance against established benchmarks, verifying its compatibility with the specific sequencing platform, and conducting rigorous quality control checks on the prepared libraries. This aligns with best professional practice by ensuring that the new workflow meets predefined performance specifications and regulatory requirements for diagnostic testing. Specifically, it upholds the principles of analytical validation as mandated by regulatory bodies overseeing laboratory testing, ensuring that the method is fit for its intended purpose and produces reliable results. This systematic approach minimizes the risk of introducing errors or biases into the diagnostic process. An incorrect approach of immediately adopting a new library preparation kit without thorough validation poses significant regulatory and ethical failures. Firstly, it violates the principle of analytical validation, which requires documented evidence that a new or modified test method is accurate, precise, and reliable for its intended use. This failure can lead to the generation of erroneous diagnostic results, potentially causing patient harm through misdiagnosis or inappropriate treatment. Ethically, it breaches the duty of care owed to patients by not ensuring the highest possible standard of testing. Another incorrect approach, such as relying solely on the manufacturer's claims without independent verification, also constitutes a regulatory failure. While manufacturers provide performance data, laboratories are responsible for demonstrating that the method performs adequately within their specific environment and for their specific patient population. This independent verification is a cornerstone of laboratory quality assurance and regulatory compliance. The professional decision-making process for similar situations should involve a risk-based assessment. This includes identifying potential impacts on patient care, evaluating the scientific validity of proposed changes, and understanding the relevant regulatory landscape. MDTs should always prioritize patient safety and data integrity. When implementing new technologies or workflows, a structured validation plan, adherence to established quality management systems, and consultation with relevant stakeholders, e.g., laboratory directors, quality assurance personnel, are essential steps. This ensures that decisions are evidence-based, compliant, and ethically sound, ultimately safeguarding the quality of diagnostic services provided. Question 15. Assessment of a molecular diagnostics technologist's actions when a rapid molecular test for a highly infectious pathogen yields a preliminary positive result, but the laboratory's standard operating procedure requires confirmatory testing, which will take several hours to complete. The technologist is aware that the preliminary result, if released, could trigger immediate isolation protocols and significant patient anxiety, while the confirmatory test might yield a negative result. Option A. Release the preliminary positive result immediately to the ordering physician, noting that confirmatory testing is pending. Option B. Contact the ordering physician directly to discuss the preliminary positive result and request their guidance on how to proceed before the confirmatory test is complete. Option C. Report the preliminary result as negative, assuming the confirmatory test will likely be negative, and to avoid unnecessary alarm. Option D. Withhold the preliminary positive result and await the validated results from the confirmatory testing before reporting anything to the ordering physician. Correct answer is A. Release the preliminary positive result immediately to the ordering physician, noting that confirmatory testing is pending. Explanation. Scenario analysis. This scenario presents an ethical dilemma common in molecular diagnostics, where a technologist discovers a discrepancy between a rapid molecular test result for a highly infectious pathogen and a subsequent, more definitive but delayed, confirmatory test. The challenge lies in balancing the immediate need for patient care and public health containment with the accuracy and reliability of diagnostic reporting. The technologist must consider the potential consequences of both premature and delayed reporting, navigating patient safety, laboratory protocols, and professional integrity. Correct approach analysis. The correct approach involves adhering strictly to established laboratory protocols for result reporting and validation. This means not releasing the preliminary, unconfirmed result for the highly infectious pathogen until the confirmatory testing is complete and validated according to laboratory standard operating procedures, SOP. This aligns with regulatory requirements for accurate and reliable diagnostic reporting, ensuring that patient management decisions are based on definitive findings. It upholds the principle of patient safety by preventing potentially erroneous interventions or unnecessary alarm based on preliminary data. Furthermore, it maintains the integrity of the laboratory's reporting system and the technologist's professional credibility. Incorrect approaches analysis. Releasing the preliminary positive result immediately without waiting for confirmatory testing is a significant regulatory and ethical failure. This bypasses established validation procedures, potentially leading to incorrect patient management, unnecessary isolation measures, and the spread of misinformation. It violates the principle of accurate reporting and could have serious public health implications, especially for a highly infectious pathogen. Reporting the preliminary result as negative, assuming the confirmatory test will be negative, is also a critical failure. This constitutes falsification of results and a direct breach of professional ethics and regulatory mandates for honest and accurate reporting. It endangers public health by failing to identify and contain a potentially infectious individual. Contacting the ordering physician directly to discuss the preliminary result and request guidance before it has been officially validated and reported through the laboratory's established channels is an inappropriate circumvention of protocol. While communication is important, it must occur through approved reporting pathways to ensure all relevant parties are informed with validated data. This approach risks inconsistent information dissemination and undermines the laboratory's quality control processes. Professional reasoning. In situations involving potentially critical or infectious disease results, professionals must follow a structured decision-making process. This involves 1. Identifying the critical nature of the result and the pathogen's infectivity. 2. Consulting and strictly adhering to the laboratory's SOPs for this specific test and pathogen. 3. Recognizing the importance of confirmatory testing for definitive diagnosis. 4. Prioritizing patient safety and public health by ensuring results are accurate and validated before dissemination. 5. Communicating any deviations or issues through the appropriate supervisory channels within the laboratory. Question 16. Comparative studies suggest that minor adjustments to PCR buffer composition can significantly impact amplification efficiency and primer-dimer formation. A molecular diagnostics technologist is tasked with optimizing a PCR assay for a novel pathogen to improve its sensitivity. Which of the following approaches best aligns with regulatory requirements for assay optimization and validation in a clinical laboratory setting? Option A. Increasing the template DNA input by a factor of 10 to ensure maximum amplification, without assessing the impact on specificity or potential for inhibition. Option B. Replacing the existing enzyme with a commercially available hot start enzyme without performing any comparative studies or validation, assuming it will inherently improve performance. Option C. Adjusting the annealing temperature and extension time based on personal experience and anecdotal reports from colleagues, without formal validation or documentation of the specific parameter changes. Option D. Systematically varying individual PCR component concentrations, e.g., MgCl2, DNTP, primer concentrations, and performing rigorous validation studies, including limit of detection and specificity testing, with all changes meticulously documented in a laboratory notebook and subsequently incorporated into updated standard operating procedures after review and approval. The correct answer is D. Systematically varying individual PCR component concentrations, e.g., MgCl2, DNTP, primer concentrations, and performing rigorous validation studies, including limit of detection and specificity testing, with all changes meticulously documented in a laboratory notebook and subsequently incorporated into updated standard operating procedures after review and approval. Explanation. Scenario analysis. This scenario presents a common challenge in molecular diagnostics, optimizing PCR for improved sensitivity and specificity without compromising the integrity of the assay or introducing regulatory noncompliance. The technologist must balance the need for enhanced performance with adherence to established protocols and regulatory requirements, particularly concerning validation and documentation. The pressure to achieve faster turnaround times or detect lower analyte concentrations can lead to shortcuts that undermine the scientific rigor and regulatory standing of the laboratory. Correct approach analysis. The correct approach involves a systematic, data-driven optimization process that is fully documented and validated according to established laboratory procedures and regulatory guidelines. This aligns with best professional practice by ensuring that any changes to the PCR protocol are scientifically sound, reproducible, and do not compromise the accuracy or reliability of diagnostic results. Regulatory frameworks, such as those governing clinical laboratories, e.g., CLIA in the U.S., or equivalent national regulations, mandate that all laboratory procedures, including modifications and optimizations, must be validated and documented to ensure quality and patient safety. This systematic approach minimizes the risk of introducing errors, ensures consistency, and provides a clear audit trail for regulatory review. Incorrect approaches analysis. Implementing changes based solely on anecdotal evidence or without rigorous validation poses significant regulatory and ethical risks. For instance, altering reagent concentrations or cycling conditions without documenting the rationale, performing appropriate validation studies, e.g., limit of detection, inclusivity/exclusivity testing, and obtaining necessary approvals can lead to a finding of noncompliance during laboratory inspections. This failure to follow established quality control and validation procedures violates fundamental principles of good laboratory practice and can compromise patient care by generating inaccurate results. Furthermore, relying on unvalidated modifications can lead to inconsistent performance, making it difficult to troubleshoot issues and potentially leading to the release of erroneous patient reports, which is a serious ethical breach. Professional reasoning. Professionals in molecular diagnostics should approach optimization by first understanding the specific performance issue or goal. They should then consult existing validated protocols and relevant literature. Any proposed modification should be clearly defined, with a hypothesis for its expected outcome. A detailed experimental plan for validation, including appropriate controls and performance metrics, must be developed. All experimental steps, observations, and results must be meticulously documented. Any successful optimization must undergo formal validation and be incorporated into the laboratory's standard operating procedures, SOP, with appropriate approval before routine implementation. This structured, evidence-based approach ensures both scientific integrity and regulatory compliance. Question 17. Stakeholder feedback indicates a need to improve the interpretation of complex next-generation sequencing (NGS) data for identifying novel molecular targets in oncology. As a molecular diagnostics technologist, you have generated a significant dataset and utilized a standard bioinformatics pipeline for variant calling and annotation. Which of the following approaches best reflects professional best practice for presenting and interpreting these findings to the clinical team? Option A. Sharing the raw sequencing data files and the bioinformatics pipeline scripts directly with the clinical team, expecting them to perform their own interpretation without additional guidance. Option B. Providing only the output from the automated bioinformatics pipeline, assuming its algorithms are infallible and require no further human interpretation or validation. Option C. Presenting a comprehensive report that includes the raw variant calls, annotated variants with functional impact predictions, and a curated list of variants deemed biologically plausible and clinically relevant, supported by literature review and expert consultation. Option D. Focusing the report on variants that align with the initial research hypothesis, downplaying or omitting any findings that contradict it, to maintain a consistent narrative. The correct answer is C. Presenting a comprehensive report that includes the raw variant calls, annotated variants with functional impact predictions, and a curated list of variants deemed biologically plausible and clinically relevant, supported by literature review and expert consultation. Explanation. This scenario presents a professional challenge due to the critical nature of molecular diagnostic data and the potential for misinterpretation or misuse. Ensuring the integrity and accurate analysis of this data is paramount for patient care and regulatory compliance. The technologist must navigate the complexities of bioinformatics tools and their outputs, understanding that these are aids to interpretation, not definitive pronouncements. The correct approach represents best professional practice because it emphasizes a multifaceted validation process. This involves not only understanding the bioinformatics pipeline, but also critically evaluating the biological plausibility of the results in the context of the clinical presentation and known biological mechanisms. It aligns with the principles of good laboratory practice, GLP, and the regulatory requirements for accurate and reliable diagnostic testing, ensuring that conclusions drawn from the data are robust and defensible. This approach prioritizes patient safety by minimizing the risk of erroneous diagnostic conclusions. An incorrect approach that relies solely on automated bioinformatics outputs without critical human oversight fails to meet the standards of professional responsibility. This can lead to the reporting of inaccurate findings, potentially resulting in misdiagnosis or inappropriate treatment, which constitutes a significant ethical and regulatory failure. Such an approach neglects the technologist's duty to ensure the quality and validity of the diagnostic information. Another incorrect approach that involves selectively interpreting data to fit a preconceived hypothesis is a direct violation of scientific integrity and ethical conduct. This bias can lead to reporting that is not supported by the full dataset, undermining the credibility of the diagnostic process and potentially harming patients. This constitutes a serious regulatory failure, as it compromises the objectivity required in diagnostic reporting. A further incorrect approach that involves sharing raw, uninterpreted bioinformatics data with stakeholders without proper context or validation is also professionally unacceptable. This can lead to misinterpretation by individuals not trained in bioinformatics or molecular diagnostics, creating confusion and potentially leading to incorrect clinical decisions. It also raises concerns about data privacy and security, depending on the nature of the data. The professional reasoning process for similar situations should involve a systematic evaluation of the data, considering all available information, including clinical context and biological knowledge. It requires a critical assessment of the bioinformatics tools used, their limitations, and the interpretation of their outputs. When in doubt, seeking consultation with senior colleagues or subject matter experts is a crucial step. Adherence to established laboratory protocols and regulatory guidelines, such as those governing the validation of diagnostic tests and the reporting of results, is essential. Question 18. To address the challenge of ensuring accurate and reproducible results in molecular diagnostics, a technologist is tasked with selecting and implementing a separation method for isolating nucleic acids from a complex biological sample. Considering the principles of separation and the regulatory landscape governing in vitro diagnostics, which of the following approaches best reflects professional best practice and regulatory compliance? Option A. Utilize a separation method validated for a different sample matrix, as the underlying separation mechanism is generally applicable across various biological fluids. Option B. Select a separation method that has been rigorously validated for the specific analyte and sample matrix and adhere strictly to the established protocol, ensuring all quality control measures are met. Option C. Modify a previously validated separation protocol to reduce processing time, assuming the fundamental principles of separation will remain unaffected. Option D. Choose a separation method based primarily on its lower cost and faster turnaround time, without independently verifying its suitability for the specific analyte and potential interfering substances. Correct answer is B. Select a separation method that has been rigorously validated for the specific analyte and sample matrix and adhere strictly to the established protocol, ensuring all quality control measures are met. Explanation. Scenario analysis. This scenario presents a professional challenge due to the critical nature of molecular diagnostics in patient care and the inherent variability in separation techniques. Ensuring accurate and reproducible results hinges on a thorough understanding and application of the principles of separation, as any deviation can lead to misdiagnosis, inappropriate treatment, and potential patient harm. The technologist must navigate the complexities of different separation methods, their underlying scientific principles, and the regulatory expectations for their validation and implementation. Correct approach analysis. The correct approach involves selecting a separation method that is validated for the specific analyte and sample matrix, and then meticulously adhering to the established protocol. This aligns with regulatory requirements for quality control and assurance in molecular diagnostics. For instance, under U.S. FDA regulations for in vitro diagnostics, (IVD), manufacturers must demonstrate the analytical performance of their tests, including the effectiveness of separation steps, through rigorous validation studies. Molecular diagnostics technologists are then responsible for performing these tests according to the validated procedures to ensure patient safety and test accuracy. This adherence to validated methods is a cornerstone of good laboratory practice, GLP, and is essential for maintaining the integrity of diagnostic results. Incorrect approaches analysis. An incorrect approach would be to modify a validated separation protocol without proper revalidation. This could involve altering incubation times, reagent concentrations, or centrifugation speeds. Such modifications, even if seemingly minor, can significantly impact the efficiency and specificity of the separation process, leading to compromised analyte recovery or the introduction of interfering substances. This directly violates regulatory expectations for test consistency and accuracy. As outlined in CLIA Clinical Laboratory Improvement Amendments, regulations in the U.S., which mandate that laboratories must follow manufacturers' instructions for use, or have a documented validation process for any modifications. Another incorrect approach is to assume that a separation method validated for one type of sample matrix, e.g., plasma, will perform identically for another, e.g., urine, without specific validation. Different matrices contain varying concentrations of inhibitors, salts, and other components that can interfere with separation efficiency. Failure to validate for each matrix type introduces a significant risk of analytical error and contravenes the principle of ensuring test performance across intended applications, a key aspect of regulatory oversight for diagnostic tests. Finally, choosing a separation method based solely on cost or speed, without considering its suitability for the specific analyte and its known interfering substances, is also professionally unacceptable. Regulatory bodies like the FDA require that diagnostic tests are both accurate and reliable. A method that is inexpensive or fast but does not effectively separate the target analyte from potential inhibitors or contaminants will not meet these standards, leading to potentially erroneous results and a breach of professional responsibility. Professional reasoning. Professionals in molecular diagnostics must adopt a systematic decision-making process that prioritizes patient safety and regulatory compliance. This involves 1. Understanding the specific diagnostic question and the target analyte. 2. Thoroughly reviewing the scientific literature and manufacturer's instructions for validated separation methods. 3. Critically evaluating the suitability of a chosen method for the specific sample matrix and potential interfering substances. 4. Adhering strictly to validated protocols and implementing robust quality control measures. 5. Recognizing when revalidation is necessary for any procedural modifications or changes in sample types. This approach ensures that diagnostic testing is performed to the highest standards of accuracy and reliability, as mandated by regulatory frameworks. Question 19. The risk matrix shows a high likelihood of misinterpreting a novel variant identified in a patient's tumor suppressor gene sequencing. Which of the following approaches best mitigates this risk and aligns with best practices for molecular diagnostics technologists? Option A. Relying on the opinion of a colleague who has encountered a similar variant previously, without independently verifying the information or consulting formal guidelines. Option B. Classifying the variant based on its presence in a large, publicly accessible variant database, assuming any listed variant is clinically relevant. Option C. Reporting the variant as definitively pathogenic without further investigation, as it is located in a gene known to be associated with cancer. Option D. Interpreting the variant using established, evidence-based guidelines for variant classification, cross-referencing with reputable clinical databases and peer-reviewed literature, and documenting the rationale for the classification. The correct answer is B. Classifying the variant based on its presence in a large, publicly accessible variant database, assuming any listed variant is clinically relevant. Explanation. Scenario analysis. Interpreting sequencing results for molecular diagnostics presents a significant professional challenge due to the inherent complexity of genetic data and the direct impact on patient care. Technologists must navigate potential ambiguities, distinguish between clinically significant findings and benign variants, and ensure accuracy in a field where new discoveries and evolving knowledge are constant. The pressure to deliver timely and reliable results, coupled with the ethical imperative to protect patient confidentiality and avoid misdiagnosis, necessitates meticulous attention to detail and a robust understanding of both the scientific principles and the regulatory landscape. Correct approach analysis. The correct approach involves a systematic evaluation of sequencing data, cross-referencing identified variants against established databases, e.g., ClinVar, HGMD, and relevant scientific literature. This process is guided by established guidelines from professional bodies, e.g., ACNG/AMP guidelines for variant interpretation, and adheres strictly to the laboratory's standard operating procedures, SOP. This methodology ensures that interpretations are evidence-based, reproducible, and aligned with current best practices for classifying variants according to their pathogenicity. Regulatory frameworks, such as those governing laboratory quality and test validation, mandate such rigorous, evidence-driven interpretation to ensure patient safety and the reliability of diagnostic information. Incorrect approaches analysis. One incorrect approach is to rely solely on the presence of a variant in a public database without considering its reported clinical significance or the context of the patient's phenotype. This fails to meet the professional standard of critical evaluation and can lead to misclassification of variants, potentially resulting in incorrect diagnoses or unnecessary patient anxiety. Ethically, this approach neglects the responsibility to provide accurate and clinically relevant information. Another incorrect approach is to interpret a novel variant based on anecdotal evidence or personal experience without rigorous validation or adherence to established interpretation guidelines. This introduces subjectivity and a high risk of error, violating the principles of scientific integrity and regulatory requirements for validated diagnostic methods. It bypasses the necessary steps for ensuring the reliability and accuracy of diagnostic testing. A third incorrect approach is to report a variant of uncertain significance as definitively pathogenic or benign without sufficient evidence. This is a direct contravention of established variant classification standards and regulatory expectations for clear and accurate reporting. It poses a significant risk to patient management and can lead to inappropriate clinical decisions, representing a failure in professional duty of care and regulatory compliance. Professional reasoning. Professionals should adopt a systematic, evidence-based approach to sequencing result interpretation. This involves 1. Initial data processing and quality control. 2. Variant identification and annotation. 3. Rigorous variant classification using established guidelines, e.g., ACM/AM. 4. Correlation with clinical information and literature review. And 5. Clear, concise, and accurate reporting. When faced with novel or uncertain findings, the professional decision-making process must prioritize further investigation, consultation with experts, and adherence to laboratory SOPs and regulatory mandates for reporting and quality assurance. The ultimate goal is to provide accurate, actionable information that supports optimal patient care while maintaining the highest ethical and professional standards. Question 20. Cost-benefit analysis shows that a new, faster sequencing technology offers significant potential for cost reduction per sample and increased throughput. However, its validation data is primarily from research publications, and it has not undergone the same level of rigorous analytical validation as the current platform. What is the most appropriate course of action for a molecular diagnostics technologist to recommend regarding the adoption of this new technology? Option A. Implement the new sequencing technology on a trial basis for non-critical patient samples while continuing to use the current validated platform for all diagnostic testing. Option B. Recommend the adoption of the new sequencing technology based on its speed and cost-effectiveness, with the understanding that further validation can be performed after it is in routine use. Option C. Immediately adopt the new sequencing technology due to its demonstrated cost savings and research-backed performance, assuming it will translate directly to clinical utility. Option D. Initiate a comprehensive analytical and clinical validation study of the new sequencing technology, adhering to all relevant regulatory guidelines before considering implementation. The correct answer is D. Initiate a comprehensive analytical and clinical validation study of the new sequencing technology, adhering to all relevant regulatory guidelines before considering implementation. Explanation. Scenario analysis. This scenario presents a common challenge in molecular diagnostics, balancing the drive for technological advancement and efficiency with the imperative of maintaining regulatory compliance and patient safety. The technologist must evaluate new sequencing technologies not just on their technical merits, but also on their alignment with established quality standards, regulatory requirements, and the ultimate goal of providing accurate and reliable diagnostic information. The pressure to optimize processes and reduce costs can create a conflict with the rigorous validation and documentation mandated by regulatory bodies. Correct approach analysis. The correct approach involves a comprehensive evaluation that prioritizes regulatory compliance and validation. This aligns with the fundamental principles of good laboratory practice, GLP, and the specific requirements of regulatory frameworks governing molecular diagnostics. Any new technology must undergo a thorough validation process to demonstrate its accuracy, precision, sensitivity, and specificity for its intended use. This validation must be documented meticulously, providing evidence that the technology meets or exceeds established performance benchmarks and is suitable for clinical application. Furthermore, the implementation of a new technology requires adherence to established change control procedures, ensuring that all modifications are reviewed, approved, and documented to maintain the integrity of the laboratory's quality system. This approach ensures that patient care is not compromised by unproven or inadequately validated methods, upholding the ethical obligation to provide reliable diagnostic results. Incorrect approaches analysis. Implementing a new sequencing technology solely based on its perceived cost savings without a robust validation process is a significant regulatory and ethical failure. This bypasses the critical steps required to ensure the reliability and accuracy of diagnostic tests, directly contravening GLP principles and specific regulatory mandates for analytical and clinical validation. Such an approach risks generating inaccurate results, leading to misdiagnosis, inappropriate treatment, and potential patient harm. It also exposes the laboratory to regulatory sanctions for non-compliance. Adopting a new sequencing technology because it is widely used in research settings, without considering its suitability for clinical diagnostics and the associated regulatory requirements, is another failure. Research applications often have different performance standards and regulatory oversight compared to clinical diagnostics. A technology that performs adequately in a research context may not meet the stringent requirements for clinical accuracy, reproducibility, and documentation necessary for patient care. This oversight can lead to the introduction of unvalidated or inadequately validated methods into the clinical workflow, jeopardizing patient safety and regulatory compliance. Prioritizing the speed of implementation over thorough documentation and quality control checks represents a failure to adhere to established quality management systems. Regulatory bodies require comprehensive documentation of all laboratory processes, including method validation, instrument calibration, quality control, and proficiency testing. Skipping or minimizing these steps undermines the integrity of the quality system, making it impossible to demonstrate compliance and ensure the reliability of test results. This can lead to significant regulatory issues and compromise patient trust. Professional reasoning. Professionals in molecular diagnostics must adopt a systematic and evidence-based approach to process optimization. This involves 1. Identifying a need or opportunity for improvement, such as cost reduction or enhanced throughput. 2. Thoroughly researching potential solutions, including new technologies. 3. Conducting a rigorous validation study that assesses the technology's performance against established benchmarks and regulatory requirements. 4. Documenting all validation activities and results meticulously. 5. Implementing change control procedures for any new technology adoption. 6. Continuously monitoring performance and quality metrics post-implementation. This framework ensures that innovation is pursued responsibly, with patient safety and regulatory compliance as paramount considerations.